I'm really happy to be back at the Broad. This is the place I first sort of realized that there were people who understood things about the human body. My high school bio education wasn't that great, and then I'm in the last year of my PhD, and I see a talk about Neanderthal DNA, and I was like, whoa, should probably pay attention to this, and I did. And so I'm going to tell you about some of the things that I learned. So I want to start actually first by acknowledging that much of this is, almost all this is joint with Loic Royer, also at the Biohub. So the other day, I walked by my colleague Jenny's computer, and she was looking at these images. Okay, so in the, does this have a laser? Yeah, okay, so in the middle here, these are kidney slices, actually, and in the middle is a sort of typical fluorescent stain. So this is a label for actin. And you do this, right, when you want to see, we've got a bunch of cells, where's this one protein of interest? But very often that protein is like a proxy, right? It's a proxy for some organelle you're interested in. And she's been working with Shailin Mehta, one of the group leaders at Biohub, to see if you can kind of learn to recognize those structures from unlabeled images, right? So if you could just put your cell in there, don't put any fluorescent markers in, and get something like this on the left. This is with, let me see if I click yes, if I'll get. So this is F-actin, and this is a birefringence image. It's a kind of, takes advantage of polarization of light to really make the structure clear. And so what they wanted to figure out is if they could sort of just learn to predict this from that. Because then maybe you would then just image this and not need to image that, right? And then you'd save yourself some trouble. And so this is sort of the input, that's the output. You train some deep learning model to do that. And what comes out as a prediction, which was the image on the left. So Jenny was sort of a little distraught because she was working on the model with Anita. The models, if you want them, are in this GitHub repo. But they were working on the model and evaluating its accuracy. And no matter what they did, it seemed like they just couldn't get the accuracy above 80%. And there's so many things you can test with deep learning models, right? So you change this, you change that, you change this, you change that, and they just they had a limit to it. But you look at the output here, and it looks really good, right? Those are the boundaries of the cells, those are the insides, and maybe the output's actually better than the label, right? Maybe the reason you can't get to 100% is that this label here is actually a noisy label, right? The molecule just happens to have stuck in these places, could have been in other places, some weird stochastic thing, and there's no way you could have predicted that from this input. Right? So somehow not only is the information of exactly where the label went not present in the input, but also you don't want it, right? This prediction here, if you're interested in these structures, is actually better than the labels you had. And so, go ahead.
So is it just pixel level binary prediction, a bit like segmentation, or is it widely formulated as a classification?
No, it's a pixel level prediction problem. And so downstream, you might then take the output of this and run some segmentation thing or some other thing on it. But this is just a pixel level prediction. And it happens to be a deep neural net, but this is sort of a general phenomenon in which when you have noisy labels that you learn from, sometimes the prediction is better than the label. And this plays out in a kind of nice way. So if x is our data and y is our noisy label, and z is the ground truth label, then we can look. Can people see what's down here? All right, I won't go too low. So you can look at the objective function, which is something like f of x minus y squared. That's what you want to minimize. And we can just complete the square. Or maybe make it into a different square. So we can just write plus or minus z here. And if we expand this, we'll get a few terms. We'll get f of x minus z squared. We'll get z minus y squared. And then we'll get some kind of cross term. We'll get twice the inner product of f of x minus z with z minus y. OK, so this is just algebraically true. And what we're training on is the thing over here. And what's kind of neat is that the thing over here, so this is somehow the noisy label loss that we have access to. This over here is the ground truth loss, which is what we'd love to be training on. This is the variance of the noise which is in particular independent of f. And if we take expectations here, right, expectations over the data, the labels, the noise, well, then something very nice happens. So when we go to take the expectation of this term, we can actually bring it inside. So if the noise is independent from the data if we condition on z, namely if I knew what the truth was and I knew what my input was, I couldn't use, if I knew what the truth was, the way that x happens to be weird wouldn't tell me what to do here. The corruptions don't depend on the input, then we can bring the expectation inside. And so we get an expectation of x given z, an expectation of y given z, and this would be an expectation with respect to z. And moreover, so this is one assumption. Moreover, if the noises mean 0, meaning that the expected value of y given z is just equal to z, right? That the noise, if we could do this a million times, would average out, then this term vanishes. OK, this term is 0. And then we're doing really well. So to put this formally, we have random variables. We've got conditional independence, so the noise doesn't depend on the x. The noises mean 0. And then for any function f, we have this equality where the what we have access to, f of x minus y squared, is just equal to the ground truth loss we want plus the variance of the noise. This is super simple algebra, but it's pretty profound. It says that you can take arbitrarily noisy labels, and as long as the noises mean 0, then training on that is as good as training on the truth up to a constant independent of the function. OK, so now let's zoom out a little bit. Why is noisy data important? Because in biology, that's basically all you have, right? So people here are really good at building things. They build new kinds of microscopes. They build new experimental apparatuses. They do crazy things. And so as soon as you build something which can measure something better, someone says, cool, I want it to be 10 times smaller and 10 times faster and 10 times higher throughput, right? And all of a sudden, your beautiful clean images are now noisy because you're at the limit of sensing. And so the two paradigmatic examples, sort of in my mind, one is SIM. This is microtubules, actually. And each dot is a single photon emission, right? You sort of stimulate it so they all go separately. And so now you've broken the resolution limit of your microscope, and you're measuring individual photons, and now it's noisy again. Does anyone recognize what's on the left? Other than basically noise?
I think it's a galactic cloud.
Right, it sort of could be, right? So astronomy I'm going to Chicago next week to talk to the astronomy people. They also have this problem where their things are noisy because they're very far away. Your things are noisy because they're very small. This is a molecule. This is a single molecule, beta-galactosidase, and this is cryo-EM. So in cryo-electron microscopy, you're bombarding an individual molecule with electrons, and as you measure it, it just breaks apart because the electrons are heavy in comparison to the molecule. And so you get these SNRs of 1 to 10. But you have a lot of them, right? So maybe you can stitch together all these noisy things and make something a little more palatable. And the last example of really noisy data is in single-cell sequencing here. So how many of you have worked with single-cell RNA-seq in the audience? Just all right, it's half the people. So you're familiar with this. For the rest, for everyone else, though, what happens is you can now sort of do the chemistry required to do RNA sequencing on individual cells. So you kind of crack a cell open, you extract the RNA you can, you sequence it. And that's great, except that the portrait of each cell you get, right, is very poor. Right? You maybe get a few thousand molecules from each cell out of the hundreds of thousands that were there. And so if you look at a matrix of gene expression, here genes are on the bottom, cells are these little rows. Right? So there's a lot of cells. It's a very sparse matrix. And so one, there's a lot of variability here, but also when you see very faintly, right, all these gaps here, is that because these cells didn't have any of this gene? Or is it because I'm sampling at approximately 1% of the total molecules, and I just happen not to have caught any, right? So when you go to analyze this data, you have a problem that you have these super downsampled renditions of all the individuals, but you have a lot of them, right? And so with the cryo-EM data of these molecules, or the SIM data, we don't have labels here, right? We don't have any Ys. We just want to know the structure of the data we have. So it's an unsupervised, or as I'll argue today, a self-supervised problem. where you have all this noisy data and you want to like somehow see the structure, and maybe you don't know what it's supposed to look like already because you're trying to do science and discover something. So there was a beautiful idea that I saw at ICML last year. Yes?
Do you use the word unsupervised and self-supervised interchangeably, or what is the difference between calling something self-supervised versus calling something unsupervised?
So when I say self-supervised, what I mean is like I'm going to end up viewing this as kind of a prediction problem where you're trying to predict parts of yourself. Sort of, you know, okay, that's the end. Everyone go home. But like unsupervised, somehow you're like taking the whole data. Maybe you're doing an autoencoding. You're compressing it. You're trying to like make some representation of the thing. Here, I'm not going to care about representation. I'm going to try to use the full power of like predictive learning to denoise data. Okay, so this was last year at ICML, Noise2Noise, and they had like one of these like in retrospect like obvious but like also super profound ideas, which is that if you have, you know, a noisy apparatus, so like everything you do is noise limited, right? Maybe you could just use it twice. Like maybe you could take, you know, your noisy measurement of a thing and then just measure it again. And if it's like shot noise, all kinds of, you know, independent noise, then you just have these two independent measurements. And of course, you could just average them, right? If you could do a super long exposure, that's just averaging. Maybe that might destroy your molecule or things might get blurry. But they had a different idea, which was just treat the second noisy measurement as a noisy label for the first one. And you know, if this sort of thing we wrote out works, which it sort of does because it's 2 lines of math, then somehow if you try to predict the second noisy one from the first one, that's going to be equivalent to learning to predict the clean image, plus some, you know, variance that you have no control over and no dependence of f on. So you do this, and they show actually they're also using a neural net that you wouldn't need to, that if you train on synthetically noised data, the performance curves training with clean targets, where you actually had ground truth to work with, and noisy targets, where you only had noisy pairs, delivers exactly the same performance. Which is cool, because even, you know, you see something like this, and you're like, wait, these are really the same? Like, that seems sort of too fantastical. But they are, you know, in expectation. And in reality, you know, training with a noisy copy as the input, as the target, is as good as having a clean one. Yeah.
This assumes a white noise, right? Uncorrelated.
No. So this just assumes what's here, which is that if I knew the true image, then the noise in the first one and the noise in the second one are uncorrelated. But you could have brown, pink, whatever terrible, you know. In fact, in this paper, they do a thing where they have text. They just stick text all over them, but in different places. And so for them, it's, you know, the structure of the noise doesn't matter. You can't look, if the whole thing turns white, right? You know, if everything is like you know, boosted, then you can't fix that, right? If it's consistent. But if it's mean zero, you're okay. Okay, so I thought this was really cool. I was like, well, we've got all these like instruments that are super noisy, but we could like take a microscope, take 2 really fast images in a row. You could start getting going here. But then I was thinking, like, what's really going on? And I like had a moment where I was like, all right. So, you know, clean data, that's fine. If your data's clean, no problem. But then, like, what if you have clean labels? Then you can train. But if you have noisy labels, you can still train and get the right answer. And then I was like, what if you are your own label, right? What if you have noisy data and you're your own label? And that would be self-supervision. So let's think about why actually, you know, data could be its own label for a second. Actually, before we think about that, we'll just write it down. Okay, so suppose you predict x from itself, right? So great, we'll just do that. X is its own label. What will happen? Like if you train on this, what happens? Yeah, you learn the identity. You're like, I know what to do. Like, nothing. Like I did nothing and it's great. And so the literature on, I'd say, unsupervised learning, which is like use an autoencoder, use PCA, something like this, what you do is you insert some bottleneck. You cripple F in some way so that it can't learn the identity function. You say, OK, it's got to be low rank. And like whatever you manage to get out of that like will be some better representation. And so you sort of like dial up or dial down the rank to control how much noise you're letting through. And you pick some nice amount, and like that's your autoencoder. But yeah, fundamentally, if you do this, you're just going to learn the identity function, right? That's what you asked for. So that's not quite good enough. But with self-supervision, take your image. This is a clean image. It's the guy. Oh, I should have said that's at MIT. This like old camera photos like over there, and it's the dome and stuff in the background. That's the dome. It's right in there. OK. What?
That's the camera.
Yeah. Oh, you're right. That's the camera. Where do you have to go to get the dome?
Like a little bit down.
All right. We'll get to the dome later. You're right. Good comment. That's not the dome. It's like his dome. All right. Yeah, very bad label on my part. All right. OK. So if you have like a clean image, it's super highly structured, right? Like, so an image of like 512 by 512 is like 250,000 pixels, and they're very correlated, right? So if I remove these pixels, which I'm calling J is just some subset of the pixels, if I cover them up, like you could fill them in, right? Like they're probably black in there, and like they're sort of like lighter in there, and it's really light in there, and like you could, not perfectly, right? There's a little information, but like basically, there's enough information in the original image, enough redundancy or correlation, you could just do that. You could just fill in from in the ground truth, from the missing pixels, you could fill in the rest. OK, fact. All right, sort of fact 2, here's the noisy image. And here's the noisy image minus those pixels, right, minus J. And sort of if you believe in the power of like, denoising of like maybe of deep learning or of classical denoiser, just anything, you might think you could recover from x minus j, you could recover z minus j. Like you don't know how to do it, but there's some function that could take you from here to here. And we agreed you could get from here to here, which means there's like from an information perspective, you should be able to get from the x without those pixels to the true values. We don't know how to yet, but like we somehow there's enough information to get there. We just have to learn what to do. Of course, what we have access to is these pixels, the noisy values, which were here. But if we train something to go from here to there, because now if the noise is independent in the different pixels, then we'll literally just learn something which does this. Right, by the same math. These pixels here are just noisy labeled versions of those, and I'm predicting them from this. Okay, that's the idea. So that's a sort of prediction problem version of this. Now, because I'm trained as a mathematician, I want a definition of like what's really going on here, and so I'm going to define a class of functions. So what we'll do is we'll take all the dimensions, all the pixels, and partition them, OK? So into groups. And we'll say a function, any function, right, from images to images here, is J-invariant if its output in these coordinates, the J coordinates, does not depend on its input in those coordinates. So for example, this says that its output in pixel j doesn't depend on the input in pixel j. It depends on everything else. You can use anything you want, but like not literally that thing to predict its own value. So, and it's J-invariant, like scripty J, if it is J-invariant for each of these. So it just says, look, you've got a function, turns images to images, and the only rule is like you can't use this to make that. You can use everything else. Go ahead, Larry. It is a subset. So if there's correlation structure in the noise, for example, you might want to split it into bigger groups. What we'll get to is the appropriate theorem here, or proposition, which is that we have now just x and z. There's no y anymore. We've removed the labels. They're random variables, and we've got our partition. And what we need is conditional independence. So if I know the true image, then the values you happen to see here and that you happen to see here are independent, right? the noise here and the noise here aren't related, conditional on reality. There's not like some conspiracy where you saw extra photons here and therefore some extra ones jumped up there. And then also the noise should be mean 0. And then if you have a J-invariant function, you get the equality that you want. You get this self-supervised loss, so f of x minus x squared is equal to the ground truth loss plus the noise variance. The identity function is decidedly not J-invariant, and the identity function xj, f of xj only depends on the value in those pixels. And here we say that's absolutely forbidden. Okay, so this equation is great because we have this. Right? We get to control what f is. We get to design our own functions. And we have the data. We didn't have z, but it turns out that we can now learn something about which function does a good job with the ground truth loss from this self-supervised loss.
Can you just remind us what are these expectations that you respect?
That's a good question. So I would say the expectations means will hold for a bunch of answers, but maybe the key one here is for x conditional on z. So you could fix the true image, and then imagine many iterations of sampling what the noise would be. And these will be equal. The cross term here, of course, will not literally be 0, but in an image with 250,000 pixels, the law of large numbers says that when you have these are mean 0 terms times another thing, and that will have some 1 over root n decay. So these will be extremely close. If we remove the expectations but had a high dimensional data, this would almost hold on the nose.
And is that assuming that the noise is in any way additive, or is there?
No. So an example of multiplicative noise, for example, is fine. Shot noise is fine. Yeah.
Is there an easy way to infer, approximate initially whether the conditional independent assumption holds? Because I can think of several cases where the noise is very much just a function of the signal. Like in images, for instance, JPEG or lossy compression noise is hugely a function of the input. In DNA sequencing, the noise profile is hugely a function of the sequence context.
So that's great. That's totally fine. You can have input-dependent noise. And so that's why the key thing here is that this is a conditional statement. It says that for fixed input, exactly what it is everywhere, then the noise is independent. So if, for example, if you're looking at a CG-rich region of the genome, right, which will be sequenced at a different efficiency, then that's OK. The issue is just if it turned out, we'll get to a single-cell example later, where basically if you had a bunch of rounds of PCR in the middle, so something had a PCR jackpot, that would cause there to be dependence between your reads in a way that didn't disappear when you conditioned on the genome. So that kind of oh, this thing happened, so that thing will probably also happen to have happened, that's bad. But the fact that it's signal-dependent noise is fine. OK. So I want to give a very simple example of a J-invariant function. Again, it's the same image. There's the noisy, there's the ground truth. And I'm going to look at a filter. So I just take a little region and I average. I take a median. And I could pick a really big region and I'll get a blurry output. I could pick a very small region and I'll keep some of the noise. And the question is which of these is best? Which of these is closest to the ground truth image? Let's parse this figure. So there's two kinds of medians we'll take. One is where we take the median of everything in some disk. And in the other one, we exclude the center. So if we exclude the center, then the output won't depend on the input there. It'll be J-invariant. And what's kind of cool is that for the classical one, right, this is the ground truth, and it goes down, and then it's good at 3, and then it gets worse. That makes sense. The same's true for the donut. It's a little worse here because we don't use the middle pixel. But then it gets better. Once we're using a 9x9 thing, right, it's pretty much the same as if we use the middle pixel. But if we look at f of x minus x, which is the self-supervised loss, that only gets worse when the radius gets bigger, right? Because if you could use the middle pixel, just do that and don't do anything else. And when you get bigger, it starts degrading. But if we use the self-supervised loss on the donut, which is J-invariant, we get this curve. This dark blue curve, which we have access to from the noise, is just equal to the dashed one, up to a constant, which is the variance of the noise. So this noise variance here is just the vertical distance between the two blue curves. And so if you wanted to, for example, tune this filter, you could use the self-supervised loss on the J-invariant thing and find its minimizing value, and that would just give you the optimal value for that function. So the way I like to think about this is that in exchange for giving up just a little bit of information, like what that pixel literally said about itself, you get totally optimal calibration. And that's often a good trade-off, because you're just guessing the value here. For the orange one, like, what are the chances what you got would be better than the tuned value for the other one? OK, but we can do a lot better, right, than like a median filter with one parameter. So here's the whole image, and here, indeed, is the dome. 250,000 pixels. And that's a lot of signal, right? We can train a complex model if we have 250,000 training points. We could train a model like this. So this is a UNet, right? It's a neural network architecture. It starts with something the size of the image, and it sort of does that autoencoder thing and backup where it compresses things. But also, it just, unlike an autoencoder, it just passes through information at the same resolution. And that's really good, because if you want to construct fine details, you don't want to have compressed your image down, right? So it's a nice harmonious thing where you get your compressed high-level representation and you can use it, but you can also get that super fine-scale detail. In particular, this thing can definitely learn the identity, right? Just pass everything through. But if we just modify it with a little masking trick I can get into more later, it will become J-invariant, and then we can just train it. And so let's train a deep neural network on this one image. And it's like pretty good, right? In fact, I compare this to like all the classical blind image denoisers, things like non-local means, BM3, this stuff. And this does better than them, right, on a single shot on one image. Already that's kind of nice. It tells you that there's something in this perspective. But the real magic happens when you go not from having one image that you want to denoise, right, but like you ran an experiment, right, and it's very badly noisy. And this is sort of my like thought experiment for that. OK, so here's some data, synthetic data. It's about 5,000 of, actually, did anyone recognize what this is? Yeah? Yeah, it's Chinese characters. OK, so there's about 5,000 of them. And I'm showing 4 of them here, but like we train on the whole data set in this. We just have noisy stuff, though. So we do this thing where we use a UNet, and we try to predict missing pixels, and we see how we do. But before that, like humans, very good denoisers, especially if they speak Chinese and can read it. So does anyone recognize these characters? Can you just make them out? Yeah, OK. Which ones? OK, so the last one's recognizable. Good, and this little bit, not really. OK, cool. So let's train. So that's like better, right? Yeah.
Quick question. Was that trained on a single image, or are there many images of the same sample?
So there were in this case 5,000.
Of each of those?
No. 5,000 characters total, no repeats. So it's learning that there's common structures in here, right? For example, it's definitely not secretly finding the ones that are the same and then averaging them, because it doesn't even have access to ones that are the same as each other. So this is cool. Like, I mean, what's interesting is like if you do, sort of recognize vaguely what's going on, like I think these are characters. So this is like, imagine you do an experiment, right? And you're like, OK, cool, these are cells. I think those might be like microtubules, but I'm not sure. And these like might be aligned, right? Like in this region, that's the equivalent of recognizing this. But like, you don't necessarily want to then like take your model of microtubules and go fit it, right? because then that will recover structures that you know they're sort of supposed to make. What you'd like to do is do something which is unbiased and that could like deliver to you surprises. Yes?
Are there any examples that it gets wrong in a surprising way?
In a surprising way, I think it gets things wrong in a maybe an unsurprising way. So here's something, right? Like this right there is an artifact. It's an understandable artifact because of that. And you know, in some sense, oh no, I didn't cap this. Oh, lucky. Okay. So in a sense, like what do we actually learn to do? What we actually learn to do, sort of f, like the optimal thing here as a function of x would be the expected value of z conditional on x. And maybe if we're doing the J-invariant thing, right, it would be conditional on the remainder of the image, right? So the best you could do from those pixels predicting the inner ones. And what this means is that sometimes if you see, I mean, humans actually do this a lot, right? That thing where like we will hear like a voice and some static, or you'll see a pattern in the rocks. And like that's because you have an inference machine and you see some noise which like kind of looks like something you recognize, and then like you see it there. And like that's what optimal inference looks like, right? Very occasionally you will see patterns in noise, and that's what happens here. So occasionally the noise will sort of like cluster up, and like it will recognize something which sort of makes sense given how the real data can look, you know, in the noise, and that totally happens.
So there was a paper about deep, it's called deep image priors.
Yes.
It's very similar to that. So what do you think about the denoising coming from the architecture itself, not the like J-invariant, or maybe the same equivalent family?
Yeah. So there's a paper called Deep Image Prior, and it makes the following observation. So if you take a generative model of images, a deep generative model, and you just start training it to produce the image you were handed, right? Single target, one noisy image, and you just train the network all 1 million parameters with that target, then it will begin to denoise that image, and then like eventually overfit. But if you like stop it soon, like the first thing it learns to do is make that image. I think that fits into this like larger history of patch-based priors for images, where something very characteristic of images is that like there'll be little pieces in them that look the same, right? If you're going along, you know, someone's shirt and there's a pattern on it, right? The same pattern will appear in many places, right? If there's a ridge, then like little bits of the ridge look the same. And so if you have these convolutional output filters, the first thing they'll learn to do is to make the patterns that repeat. Which is to say that like convolutional nets are biased towards features of natural images. In this case, I think what's going on is that like, you know, a non-convolutional net, like argmin is the right thing, right? If I had infinite data, it would learn to do the right thing. But if you come in with something with good biases, like image biases, then you'll learn it a hell of a lot faster.
Do you use convolution?
So I'm using convolution here. And in general, like with deep learning or with any learning, right, like if you don't have a prior, you need a lot of data. And if you have a good prior, you need less data. And so embedding convolutions in it is good. For all of those of you who work on like gene expression or the genome, right, what makes it so much harder than image processing is that we don't have good priors yet. Like we know what neighboring pixels are, and maybe they're related, but we don't know what like neighboring genes are very much. And so you go into those with much weaker priors, and it becomes much harder to train. What am I going to say next? OK. This is from Loic and Chinmay, and it sort of gets to why self-supervision might be desirable, even if you had training data you could use. Like, even if you could train on clean stuff, here's why you might not want to. And that's that data-driven image reconstruction, I don't know if it precludes discovery, but it's very troubling. So here's the example. Here's a degraded image. And they consider what a net does on this, a net that had been trained in 4 very different regimes. In the first regime, it was trained not on this word, but on other words in English with all the letters present. And you run it, and you're like, that's pretty good. You know, maybe the T could be better. In the second case, it was trained on a bunch of words, but those words didn't have A's or E's. So like pretty good, right? In biology, you're not going to get better than like your training data was missing some A's and E's, right? But then you go and like there aren't A's and E's here. right? It put in a good inference, right? If you only knew about G's, then that's like kind of closest to a G, and it'll just stick it in. And that's the right thing to do from the perspective of the training loss. If you train it on Chinese characters, you get this, which kind of looks like biology. Right? Like kind of a mess, but like maybe there's something there if you squint. And like most troubling to me actually is this last one. So if you train on something which has the whole English alphabet, but also the Hanzi characters, then it degrades the quality of your output because you might think that you're seeing some of the Hanzi characters, right? And so it's a little worse because you have these alternatives. And so what's nice about self-supervision is that you can actually train it on the one experiment you did, right? If you can train it on one image, you can definitely train it on like a stack over time. And so you don't have the problem of biasing yourself towards the training data because you are your training data. And then last on the image thing, and then we'll move to single-cell stuff. I wanted to shout out to Alex Kroll and Florian and Timo, their work on this. So, you know, I was not the only one to see like Noise2Noise come out and be like, that's great, but what's really going on here, and have that like sequence of brain explosions. And so right before we put our manuscript out, they put one out that was doing the sort of deep learning part of the story, where they did the sort of swapping out of pixels, some minor deviations that if you're really interested in, you can like read the supplement to my paper to get into it. But I thought they had a really nice example of what happens when independence fails. Okay, what happens when your noise is structured? And so they do this for two examples. One is like they add structured noise on top of this boy. And so there's like the random part and the structured part. And when you denoise, like you can see that pattern's there. And that's because if you use pixel level independence assumptions, then you could totally predict the grid noise from that, and lo, you keep it. And so in some sense, my definition of noise is like the stuff you can't predict from the other parts. And if you want to dial that, you can change the region of independence, right? You can like use more pixels, or we'll get into the molecule case. And then they also did this on some microscopy data where you know, they didn't add it, but and so there's no clean target, but like they get this very faint gridded pattern out, right? And that's telling you your microscope has some systematic errors. Those not only will those persist, like you'll really see them, right? Because the rest of the noise will disappear, and you'll be left with the structured stuff. And if you want to get rid of structured noise, you know, you have to do that yourself, right? Like no self-supervised, unsupervised, whatever method can get rid of structured noise because it sees it as structure, and it's like, congratulations, like here's the structure you were looking for. And so if you have some opinion about which structure you want and which structure you don't, like you need to go say that in some way. OK. Now for something completely different. Oh, not for something completely different, but rather for a question.
Yeah, before we go on, do you have anything to say about, like, I realize you can do it on a single image, but the amount of data, say the size of the image compared to anything about F, like how complicated F is? Is there any, have you explored that?
Yeah, so it, you know, you should have, you know, probably as many data points as parameters, but a data point for an image problem is a pixel. So you always have, you know, as soon as you have 10 images, you have more data than parameters in even a super deep net. And so the real question becomes like, so there's no architecture constraints really based on data. I'd say your performance tends to get more magical the more data you have, the more information you can pool. And so when I do the Chinese character thing, but I only use like 8 of them instead of 1,000, it's better. It's like better than the classical stuff. But that thing where it just like crystallizes in starts to happen when you have more.
And anything you're regularizing in there at all, or just in normal way? No, nothing at all, other than the architecture of the model?
Yeah, nothing. You don't need to because here actually the self-supervised problem, it's the one you want to solve. The issue with like the one where you can learn the identity is you don't want to solve it, so you have to add a ton of regularization. But here, like, it's desirable to minimize this function. OK, this is joint work with James, who's also at the Biohub. OK, yes?
So going back to the microscopy experiment. So practically speaking, then, what you need to do is capture at least a few images that aren't compressed. For example, if your microscope only spits out JPEGs, you're always going to produce JPEG artifacts if that's the only thing you have to train on, right? Or
yes. Assuming that, I mean, so JPEG artifacts, what are they? They're like this blocky texture, which is super spatially correlated. Now, if you want to get,
If you had like a few non-JPEG, like if you captured 10 TIFF with no compression and the rest JPEG to save space, you could learn from the TIFF, right?
let's get into that question. Let's get into that question later. In the JPEG case, you might even be able to get out of it if you work in the wavelet basis, which is the natural basis which is giving it that blocky structure. And there might be independence between the wavelet coefficients. But let's do that in the question session. So I want to get to single cell. As I said before, you have individual cells with like maybe a few 100,000 molecules, and then you manage to capture with your sick drop-seq system like thousands of them, very lossy, and that affects all your analyses. So one way this shows up is if you have genes that like in a deeply sequence, this is like 40,000 molecules per cell, you can see there's some strong correlation here between these two genes. This is HCA preview data set, and also there's some outliers here. Right, there's both some like subgrouping and a correlation. And this is much more typical. This is what like the data actually look like that come out typically. And here each point is a cell. These are the gene axes, and I've jittered them so you can see that like most of them are at 0, 0. But this is what it is. And the correlation here is like 0.03 instead of 0.24. So like this is what you see from your experiment. This isn't even what was going on. This is like closer to what was going on. This is like factor of 10 from reality, and this is factor of 100 from reality. So this is where you're at. This is the Paul et al. Data set, and it's what I showed you before, right? This is the whole, actually a part of the expression matrix. So genes here, cells here, very sparse. Okay. So we want to somehow use the same idea. One thing you could do immediately is just treat this as a matrix and just drop out entries, right? Predict some entries in this matrix from others, and that'll work. But here we can take advantage of a somewhat deeper structure to help us learn how much information to pool. And I'll tell you about that right after I show you the hazard. So, one thing you can do with a lot of noisy data is average, right? That's a guaranteed way to reduce noise is averaging. And if you do your single cell experiment and you average all of it together, you did an extremely expensive bulk experiment. But no problem, right? I mean, like somehow that average is a good population average, and you did it. You could also leave all the cells separately, and it's very sparse. But maybe you want something in between, where you share some information to get substructure, but like not so much that you've smoothed the whole thing out. And the simplest way to do this is with something like PCA. So here's the raw data, and what you could do is project down and project back up. So here's what happens if you use 3 PCs, or 9 PCs, or 100 PCs. And you look at these and you say, indeed, this seems smoother as a relationship between these 3 genes. The third one's in color. This is somewhere in between, and this is like pretty coarse. And you could try to figure out what to do by looking at these scree plots where you see the norms of the principal components. And like maybe, where do you pick it? You pick it somewhere on there, right? But like this has that autoencoder issue where like obviously the more of these you include, the more variance you explain in your data. And if you include all of them, nothing's happened. Anyone have an opinion of like how many of these to use? You should always pick the middle one, right? That's the point. It's not enough, like too much in the middle.
Neither one. 0 is pretty good.
Yeah. So then you might say, like, this is a terrible model using PCA. Why are there negative numbers here? And that's a reasonable objection. Okay. All right. So here's the way we'll use independence in this context. Okay. So what happened? We had the cell with a bunch of stuff. We detected a few. Let's split those up into 2 groups. So we take all the molecules we saw and we split them into 2 groups. And as long as there's no like bias in the bead where like I got this and therefore I got that because they're sort of like physically attached to each other already, then we can view this as being 2 independent samples from the original cell. Like you had 1 bad sample, now you have 2 very bad samples, but they're at least the only thing they have in common is they came from the same place. And that means that if we do something to this one and compare it to that, it's a noisy label situation. This is a noisy label for that, and the ground truth is the original cell. So by somehow comparing what we do on this to that, we can get an estimate of how well we're recovering what was there originally. So in the language of the matrices, here I've taken the previous matrix and I've split it, taking one molecule and tossing it to each side. Right? Now I have two matrices. As you can see, they're quite similar, right? Same cells, but they're not identical. Especially in some of these sparser columns, there's intensity differences. And where it's really sparse up here, just different rows will have had stuff in it, right? This one got the molecule I measured, and like that one got the other one. And so, indeed, for very highly expressed things, like if you saw 100, then there was a lot, right? But if you saw 1, then maybe there were 2, and maybe there were 7. And if you saw 0, maybe there was 1 or 2, right? And so in that low regime, this has a dramatic effect. And now what we can do is we can apply any denoising function we want, PCA, diffusion, like deep neural nets, whatever you like. On the first one, we can compare it to the second one. And this squared loss, as before, will be an estimate of how well we're actually doing. So we do it. So this is two different methods. This is PCA, and this is like a diffusion method where you sort of, yeah.
Just a question about the narrative. You talked about this having two noisy measurements, and then you showed a new method that you didn't keep that, and now you put it back. And I'm slightly confused why you're doing that.
Right. So the way I think about this is you have like one measurement, which is the whole cell, and we want to zoom in on the independent structure. And with pixels, we said, OK, the noise in each pixel is independent. And here I'm saying each molecule is independent. And so my J here, instead of doing like 10% of the pixels on one side, I could do 10% of the molecules. I'm doing half the molecules because it's like easier numerically.
So you basically have J partitioned into 2.
Yeah, so I'm doing a partition into 2 instead of a partition into something like 64 or whatever would be more effective.
Okay.
You need into 2 because it's super sparse already.
So there's an interesting challenge when you do this. The more you put on the right side, the better your answer will be. You have more information. But the more you put on the left, the better your estimate of the loss will be, the lower the variance. And so you can sort of dial how much goes here versus how much goes there in terms of like how powerful your model will be and like how well you'll estimate how well it's doing. Also, the honest answer is like there's a bunch of normalization questions if you're going to do like log normalization or square root or medians, whatever you're going to do, that completely go away if I do equal amounts. And if I don't do equal amounts, I have to write more things down. Yeah.
So in the image case, it seems like you haven't talked about how to select partitioning. But
Yes.
you can see, you feel like in the image case you want, if you weren't worried about computational concerns, to have every pixel be its own partition.
Yes.
And here you could do the same thing where you leave out one molecule.
Right.
Right.
So what we've been calling this is like molecular cross-validation. And with cross-validation, like the choice of like how big to make your layouts. People usually like 10%. Like you could do 1, leave 1 out cross-validation, and then you just need to do it a lot of times to estimate your parameters. And that's fine if it's a deterministic model. If it's like gradient descent, so you need to do all the crosses to take one step, then that's a bad idea. So if one way to think about it is like you could have half your information here and half there. By the time you're at 90% on the input, like you don't get that much for bumping up to 100. And you would start getting super high variance estimates. OK, but what's cool here is because we're using this self-supervised loss, f of x minus x, or in this case the split, right? It's in like the same language, the same units for all methods, right? So we can not only find that the best value for PCA is like the middle one, obviously, but also we can compute the best diffusion time for a diffusion method, and we can see that the PCA is actually performing better. So if you had to pick one just based on this reconstruction here, you'd use PCA. This is a deeply sequenced data set where I'm actually, so these are based on about a few thousand molecules. This is based on the 40,000. And this is just to show you that this gets us, these curves mirror something closer to ground truth. So this is how good the recovery of 40,000 molecules is from 3,000. And as you can see, as with the median filter before, these curves have like exactly the same shape. So when you do the self-supervised loss, you really are doing the same thing as optimizing the ground truth. John?
You say, right, with diffusion maps, PCA, I'm imagining like I get a reconstruction error because I can actually think of my data as just shoved onto some low-dimensional subspace in the same ambient space or some distance. With diffusion maps, you're Like, how are you doing the comparison? You're going down, how are you going back up?
Oh, I'm not going down. So the way the diffusion works, I'm not doing diffusion map, I'm just doing diffusion. So we'll take all the cells, we'll find their nearest neighbors, and we'll make a graph. Okay, so that's some graph structure. And now we can diffuse on the graph, where we basically average each cell with its neighbors. Right. And the question is like, how big a neighborhood do you take?
I see.
And so you could take the whole thing, at which point you have bulk. You could take just itself, at which point you've done nothing. And that's why for diffusion, this loss goes up. The more you diffuse, the further you are from the input, from the autoencoder. But you do better up to a point.
So it's a lot like a donut.
Yes. This is like the donut, basically, on a graph. Yeah. Well, it's not. So you could do the donut on a graph. This is slightly different because I've already split the molecules. You could do donut on a graph where you like literally just average a cell with its other ones, but not itself. Right. That would also work, but it's not what I'm doing. OK, what did I say next? Oh, yeah. So this is like the denoise thing. This is where I blow up the contrast in the columns so you can see the structure more clearly. It's sort of worth noting here that you need something to tell you when to stop. Because like a lot of the papers here, they say, look, like I have a block structure, right? There's clusters of cells which are similar, and now their expression looks more similar than before. And you're like, cool, you want clusters that look the same. I took your clusters, I averaged everything inside the cluster, and then I gave it back to you. Like I could make these blocks all red in those corners, right? And that would maximize the how blocky is your output metric, which is often used like as a validation that you're doing the right thing. I think of this as basically someone giving you back a super blurry image and being like, there's no noise. And it's like, yes, like there's no face. OK, and so just one example here. This is a diffusion method called magic. And maybe the question is, how much should you believe in magic? This is a video from their sort of tutorial where they show 3 genes as you diffuse longer and longer, right? So you start, it's really hard to see a relationship, and then something pops in, and then it just keeps getting smoother and smoother and smoother. And if you dial it all the way, you get this nice, beautiful little line. And the question is like, all right, I can smooth the data. How much? Right? When do I stop? And we can use the self-supervised loss, and it says stop like immediately. Like take one step, and then that's it. And like, maybe that's a little too much, but like one step was good. And so, and there's a much, there's a relationship you can see here. It's definitely, better than that, But like once we go here, this has gone too far. Right. So with the self-supervision, you can say like this is okay, good, good enough. What was I going to say? Oh yeah, what next? So, I mean, what next for me is like do my ICML revisions by next Monday and like then write up some of the Singlecell stuff, which is currently just like a tiny bit in the supplement there. But I think what next bigger is like so much of the data that people gather here is extremely noisy, and you're in a position where you might want to either just for exploratory purposes, just to see what's there, like with the Chinese characters, you might want to try denoising it. But also, if you're going to go feed it into some other step, like finding nuclei of cells or finding clusters in the single cell case or finding patterns of gene expression, right? It might be helpful to denoise first and then do something which works on relatively clean data instead of trying to build something that has to handle all of the crazy noise which is present initially. And so, the denoisers will only be like as good as the models are, right? This doesn't solve the problem. Like, it doesn't tell you what was there, right? If it gets you closer and it tells you how much you're getting closer compared to methods, and then you might be able to use that to do the next step. And with that, I want to thank a bunch of people. We've got a photo wall at Biohub. We're about 100 people, including like data science, which is my team, but also in a huge infectious disease group, bioengineering, microscopy, including Shalin, the cell atlas group, ops team, and like one of the fun things there is that like there's mathematicians like me, but also people who build microscopes and think about that stuff, people who work on dengue and those kinds of things. And so it's a fun place to sort of take an idea like this, which started with images, and then sort of bring it and see where it can go. But I think this is only 100 people, and like there's a lot of work to be done. And I hope people here will stay afterwards and chat, ask questions, and think about ways you could use similar methods on the data you're working with. And that's it.